Another type of diagnostic test is called an immunochemistry diagnostic. In this case, we are using an antibody to detect a protein, and really a protein antigen, that is specific to a particular marker for a diseased state. So for example, the protein troponin is associated with cardiac diseases, diseases of the heart. If you have a heart attack, some of the heart cells die and then release troponin into the blood. And by taking a blood sample, you can detect the presence of elevated levels of troponin using an antibody. The antibody is mixed in with a sample of blood, and then we try to detect the presence of an increased amount of troponin. Alpha-fetoprotein is a protein that becomes elevated in certain cancers. Therefore, the presence of an increased level of alpha-fetoprotein is an indication that you may have a particular type of cancer. And again, there is an antibody that we can make that will detect alpha-fetoprotein. So, just to be clear about this, for each of these different proteins, we have to produce a different antibody to detect it. So each protein or each biomarker for a particular disease has to have its own specific antibody, which we have to make in the lab. And of course, we've used antibodies to detect HIV now for a couple of decades. And the advantage of using an antibody as a diagnostic is the fact that it is exquisitely specific. It's less likely to give us a false positive test. It will recognize a specific protein, and in some cases, if done correctly, this can also give us some quantitative information. We can also use antibodies to detect substance abuse. While these are not proteins, we can detect heroin, cocaine, steroids by producing antibodies against particular small molecules. It just takes a little more work to do that. But again, these tests are highly accurate. It's difficult to question the results of an antibody test, and they can give us quantitative information if we need them to.